Hello, welcome to the Chapter 7 presentation. I want to encourage you to have your Chapter 7 reading guide out while I go through this. You can probably get most of that, if not all of that, done during this presentation. So I'm here in Chapter 7. This is all about cell membrane and movement across it. Some of this we've talked about already, so some of this I might go pretty quickly. But the plasma membrane is a boundary. Remember, we called it kind of a barrier. So it separates a living cell from basically what is surrounding it. So it is what we would consider to be selectively permeable, which means it can control what comes in or out. And we sometimes call it a fluid mosaic of lipids and proteins and even carbohydrates. And it is consisting of a phospholipid bilayer, which we've talked about this year already, and it does have proteins within it. So if we look at this picture, we can see why they would maybe call it a fluid mosaic. A mosaic meaning many parts making the whole. And you can see that that definitely makes sense for this picture. And we'll go through different parts of that picture throughout this chapter. So phospholipids are the most abundant lipid in the plasma membrane by far, and they are what we would consider to be amphipathic. So number 2 on your reading guide, amphipathic, meaning they contain two different types of piece to them. So they have a hydrophilic head and a hydrophobic tail. So we call that amphipathic. The head is composed of a phosphate group that is polar, that can interact with water, and that is can attach to a hydrophilic. The head is hydrophilic, and that is attached to the tails, which are hydrophobic, and that is the lipid part. So you've seen a diagram on my board probably that looks similar to this, but we labeled this in class last week, and we had the phosphate heads again. And again, we can see water inside and outside of the cell. And those phosphate heads, because they're polar, they have to be oriented toward the water. Polar is attracted to polar, and then these phosphate tails create the barrier. They are hydrophobic. So Singer and Nicholson are the first ones that were able to really visualize this in a freeze frame, but we're not going to get into that too much. The membrane is what we would call, is a fluid structure with a mosaic of various different things embedded in it. And phospholipids can move. Um, they do have some flex in their tail too, which allows some fluidity, allows some motion. And membrane proteins also move side to side and laterally as well. So, about how often does that happen? Well, laterally, so side to side, it happens all the time. You can see 10 to the 7th times per second, so an incredibly high amount of times. But flip-flopping, meaning going inside to outside, that happens rarely, but does happen about once per month. Now, what influences this? Several things, including temperature. Temperature is probably the most important one, and the cell membrane is much less fluid at a colder temperature. Other thing that would affect the fluidity of a membrane is the type of hydrocarbon tails. So hopefully from our last unit you can tell that these kinked ones would be considered unsaturated, and these straighter ones over here would be much more saturated. So actually the ones that are unsaturated are much more fluid. Okay, the kinks prevent the tight packing on the left, and that allows for more fluidity. Another thing that can affect the fluidity of membranes is cholesterol. Cholesterol is a really common steroid molecule that's found in the cell membrane. And when it gets really cold, it kind of acts as a cushion. And when it warms, it actually can help to restrain movement, reducing fluidity, which also can be good. You don't want it to be too fluid, otherwise it can break apart. So Cholesterol is very important to the structure, allowing for reduced movement when it's really warm and increased fluidity when it's really cold. So the membrane is really a collage of different proteins embedded in it. So it also has proteins and the main thing that these proteins are most associated with is movement across and communication across the membrane. They are specific, meaning like for instance, water is something that has to be able to go across the membrane. There is a specific protein called aquaporin that allows water across, but it only allows water. It's not going to allow like food or glucose to come into the cell. There's also peripheral proteins, and they're not embedded. They're loosely bound on the surface, mostly for cell-to-cell communication. Then you have what's an integral protein. So aquaporin that I just talked about which is the protein that moves water, uh, is one of these. And these span the membrane, meaning they go all the way across. And they do have amphipathic pieces as well. So they have to have polar parts that interact with water and non-polar parts that can interact with the non-polar piece of the phospholipid bilayer. There is 6 major functions of the membrane. The first of which is transport. Now, if you look at number 4 in your reading guide, all you have to do is list these. So the first one, transport. Second one is enzymatic activity. So sometimes they can catalyze reactions. We do also have signal transduction. I know you can't really read that very well for whatever reason. The formatting got bad, but that says signal transduction. And we'll talk a lot about signal transduction when we get into cell communication. So you can guess this is all about communication between cells. Cell-to-cell recognition, which is similar to communication, but a little bit different. Really important thing in the immune system and recognizing self versus not self cells, so they know which ones to get rid of and things like that. There's also intercellular joining, which can connect cells, maybe in like an organ or in a plant cell. That would be important. And then they also can have attachment to cytoskeleton and extracellular matrix, so that they need to fix themselves in place or fix something inside of the cell to it, and for structure and other things. That is also something that proteins can do. So the role of membrane carbohydrates in cell-to-cell recognition. Cell-to-cell recognition is cells being able to communicate with each other and recognize what is this neighboring cell. This is really important early on in development for sorting cells. So certain parts, basically if you have a stem cell, which is a cell that can become anything, if certain part of the body is becoming, say, the brain, it can interact with the other cells in that area so that they become a brain too. And other parts are becoming, say, the liver, and other parts are becoming muscles and things like that. So they know what to become. And they also are important in rejection of cells. So if, say, a bacteria that your body doesn't want in it and shouldn't be there, your immune system cells can recognize it and get rid of it. And membrane carbohydrates would come into play with that. So that really is what distinguishes, so that cells can recognize, is this something that should be here? Is it not something that would be here? A good example of that is the different types of blood. So type A, type AB, type B, and type O. What's different about them is they have different membrane carbohydrates. Membranes also have distinct inside and outside faces, and this can affect the movement of the proteins as well that are synthesized in the endomembrane system, which we talked about last chapter. So this is that review on membrane. So here we have the ER creating a protein through a transport vesicle to the cis side of the Golgi, and then the trans side of the Golgi is going to bud off of another vesicle, and you can see it's being secreted in this case. But the membrane and the sidedness of that membrane is important so that those transport vesicles can interact and bond with them. Membrane structure results in selective permeability as well. Selective permeability is, again, controlling what can go in and out of that cell in both directions. They have to be able to get waste out and nutrients and sugars and water in. So it has to be able to exchange materials with its surroundings. So hydrophobic molecules, they are considered lipid soluble and they can pass through the membrane. Now, if you're wondering why, well, remember phospholipids, the barrier on the membrane, so like dissolves like, right? So lipids can pass directly through. Those things like the steroids and carbon dioxide would be another example. Oxygen, another example that can pass freely through, but steroids like testosterone and estrogen would fit there as well. Polar molecules, on the other hand, do not cross the membrane rapidly. Polar, an example that we should be familiar with is water. Glucose would be another example, polar molecule, or salts that cannot pass freely. So what do they do? What they need is called a transport protein. So you can see in this picture what a transport protein does. If this is the polar molecule that needs to cross, it needs to go through that protein. It's basically allowing transport across that membrane. Water's protein is called aquaporin, as I said. But each protein is very specific to the substance it allows to go through. Glucose's protein won't even allow fructose, which is an extremely similarly shaped protein. So these are very, very specific. And remember why? What's the most important thing about a protein is its shape. So its shape has to be perfect to allow the molecule that it is allowing to cross. The first type of transport that we're going to go through is passive transport. If you think of passive, that word means it requires no energy. So I would underline that. If it is passive, it is not requiring energy. So things like carbon dioxide, water, and oxygen easily can diffuse across the plasma membrane, and it doesn't require any energy to have that happen. Diffusion of water is known as osmosis. That gets a special name because water is so important. One type of passive transport is called simple diffusion. And simple diffusion is the tendency of molecules of any substance to spread out evenly and move from high to low down their concentration gradient. Simple diffusion goes directly through the membrane. In other words, as far as a cell would go, this would mean it can go directly through the phospholipid bilayer. This does not require a protein. So this is called simple diffusion. No work is required to happen. There's no energy. Okay, simple diffusion. And the molecules would continue to move back and forth, and even at equilibrium, so when equal amounts on both sides, they still move. It's just no net movement anymore. So they can move back and forth at equal rates. But until they get to equilibrium, here we have, again, a high concentration to movement to a low concentration directly through the membrane. Simple diffusion. We also have osmosis, which is an example of facilitated diffusion. Facilitated diffusion requires a protein in order to cross. So osmosis is the movement of water across a semi-permeable membrane. It is an example of facilitated diffusion. It is affected by the concentration gradient of different substances and how much solute is in the water as well. So we'll go through a couple examples, but you need to be aware of these terms. The first is hypertonic. And hypertonic, if you look at this diagram, is when water is leaving the cell. Now, remember, water is a diffusion process. So if I were to grab my pen here quick, This would be my high concentration inside the cell. This is my low concentration. What am I counting for that? I'm counting the amount of water molecules. So if I have a high concentration inside, remember through diffusion, it has to move high to low. So in this case, it's going to move out of the cell. Hypertonic. All right, hypotonic is the exact opposite of that. The high concentration now is outside the cell, and it's going to move inside, which is low. Again, still moving high to low. And how I think of hypo, I think of the letter O, and it's kind of the same shape as a balloon. And a balloon gets bigger, so what's going to happen to the cell? It's going to gain water, and it's going to balloon. It's going to get bigger. Hypo. Maybe sometimes I think of hypo hippo. Hippos are fat because they get a lot of water in them. All right, so they're big. The cell is growing because water is entering. And then the last type is called isotonic, which has equal amounts of water going in and out. And that's just because there's no high or low concentration. We have equal amounts on each side of the cell. Now, if we were looking at an animal cells are most, well, I don't know if this is the right term, but they're most happy isotonic. Okay, they want to have equal amounts of water inside and outside. If we get to a hypertonic environment, our cells shrivel up, and that's not good. If we get in a hypotonic, our cells swell up with water and eventually can burst, like this cell has done. This is why it's so important when you get fluids inside of your body. You get like saline, and saline is isotonic to our blood so that this does not happen. So again, just showing that cells are happy and our animal cells are happy in that isotonic solution. Hypotonic, they can burst. Hypertonic, they shrivel up. Now, on the other hand, it's a little bit different with plants because plants rely on water for their structure, and that deals with turgor pressure. They have a cell wall made of cellulose, and that helps to maintain the water balance and help have that rigid surface. But turgor pressure is the pressure of water inside the plant pushing outward against that cell wall, and that's going to allow for some of that structure. If a plant is considered turgid, it is full of water. It is hypotonic. Remember, water into the cell for hypotonic, making that swell swell and push against its cell wall, creating that pressure. And I'm sure we've all noticed that plants sometimes are wilted. Sometimes if we go through like a little drought or something, those plants that are wilted need water. They don't have water pushing against their cells to allow their structure to stand up straight. So if we look at this diagram, it does a nice job of showing that. We have the cell on the left being turgid, and what I would say is this is where a plant cell is most happy. It has a full vacuole, and water is pushing out against its cell wall, allowing that plant to swell. You can see the cell wall is swelling out, and that pushes out and allows for structure. This would be flaccid. This would be the wilt example. This cell is fine. It's not dead or anything. It's in an isotonic environment, equal amounts of water in and out. But this plant is wilting because it's not fully pushing, that water is no longer fully pushing out against. There's not enough of that. Okay. Then if we get into the hypertonic, so hyper, let me label the other ones. This was iso and this was hyper. Okay. If we get, this is hypo, not hyper. If we get into the hypertonic, this cell is what we would call plasma-life. Now, I'm probably guessing you've all seen this and not realized it as well. What happens to your grass if it goes through a drought? It turns brown, right? It looks dead almost. Well, that grass in reality is not dead. How do I know that? Well, what happens the first time it rains after your grass looks like that. The grass greens up pretty quickly again. Why? Because your grass has plasmolysed. And basically, this is an adaptation that a plant has to survive in this low water environment. So they plasmolyze themselves to basically preserve. But again, what happened here? There's water leaving the cell. So hypo, water coming in, iso, water coming in and out at equal rates, and hyper, water is leaving the cell. All right, facilitated diffusion. Again, this is movement across the membrane that requires a protein. So transport proteins are required for this process. And we can have a couple different types. One could be a channel, which is basically just a corridor, a tunnel through. You can have a carrier, which has to move. This protein actually has to move to allow it through. And these are just a couple example problems. Let me give you a chance to read this. The solution A has 100 molecules of glucose per milliliter. Solution B has 100 molecules of fructose per milliliter. How will the water molecules move? Okay. Well, this solution, I'll give you a little bit of time to think on that. But glucose and fructose have the same molecular formula. So in this case, this is considered isotonic, and there would be no net movement of water because the concentration is equal. If we did this similar problem, we have solution A with 100 molecules of glucose per milliliter, and solution B has 75 molecules of fructose per liter. Again, glucose and fructose have the same molecular formula, so you can think of that. How are the water molecules going to move? In other words, is it going to move from A to B or B to A? Think about that for a second. And if you did that correctly, you would have a net movement of water from B to A. In other words, what we're saying is B is hypertonic to A. B is hypertonic to A. And our last one like this, solution A has 100 molecules of glucose per milliliter. Solution B has 100 molecules of sodium chloride per milliliter. How are the water molecules going to move? This is a little bit of a trick question. Give you a little time to think on that. So maybe me telling you that this was a trick question helped you with this, but a lot of students I have do this. If I don't tell them that, would say, well, it's isotonic. There's 100 molecules of each, right? But if you think about sodium chloride, that's salt. What happens to that in water? Well, that salt will dissociate and therefore create 200 molecules per milliliter. So in essence, what it's going to do is it's going to create a solution. The water is going to move from solution A to B. So B is hypotonic to A is what we would say. All right. Active transport is the last piece of transport. And the key part, if you are active, you are requiring your body to use energy. So active transport is requiring a cell to use energy. And the reason why is it's moving solids against their concentration gradient. Against their concentration gradient. In other words, what that means is low to high. So in order to move against their concentration gradient, we're moving low to high. In the previous examples, we were talking about diffusion. That was always high to low. And that we would say is down their concentration gradient, not against. So if we're going against, it's low to high. And that requires energy. And what do we use for energy? In this case, we use ATP. Okay. So very important. Active transport requires energy. And we get that energy in the form of ATP. And this is what you're seeing here. An example of that is a sodium-potassium pump, which we'll talk a little bit about when we get into the nervous system. But what you're seeing here is it's moving their solid against their concentration gradient, And here's the ATP that's being used. That's going to provide the energy, but it's going to allow that to go low to high. So this is a great diagram, a really good slide, good study tool. Star this bad boy. Good review of all the different types of transport. And when we do this, we're going to model this in class next week. This will be a good slide to reference. What you're seeing here, this is considered simple diffusion. How do I know? It's going directly through the membrane. It's going high to low. Okay. This is directly through the membrane. This is simple diffusion. Okay. Now these two are facilitated diffusion. How do I know? They require this protein. It's still diffusion because it's still high to low. Is energy required? No, it is not. It is considered passive. Now on the right side here, we have active transport. Now what are we moving? In this case, we're moving the squares or the diamonds. And what we're moving, this is a low, and here we have a high. Now where are the molecules moving? They're moving in this direction. So they're moving against their concentration gradient, which is why we need this energy. In order to move against the concentration gradient, we need energy. All right. So just to wrap it up now, membrane potential is the voltage difference across the membrane. We'll get into membrane potential a little bit when we talk about action potentials, but not really important at this point in time. A proton pump moves protons across a membrane, and that does require energy. We'll talk a little bit about proton pumps and the electron transport chain, but not really important at this time. Co-transport is an important concept, and co-transport just occurs when the active transport of a solute indirectly drives the active transport of another solid. So it's one leading to the next, and it's best shown in this diagram here. What you have here, you can see the transport of this hydrogen across, this hydrogen ion, is indirectly allowing this then sucrose to come into the cell. Okay, so that's an important concept. One movement is allowing the next movement, and that's called co-transport. And that, I believe, is, I think we got a couple more slides actually. Bulk transport is another type of active transport, movement into or out of the cell. We can have exocytosis, which exo, think exit, and endo, think in. So it's basically allowing large things to come in or out. So exocytosis, we think of secretion out of the cell, maybe through a transport vesicle. Endocytosis is taking that in. Think of like a food vacuole. No, this doesn't work anymore. This used to be an animation. But endocytosis, this cell membrane would go out like this and out like this and eventually come together and bring this molecule in. Okay, so it's bringing it into the cell there. And exocytosis, what you're seeing here is this is budding off. So this is your cell membrane here, and eventually this is going to come together, pinch this off, and this is going to be released out of the cell. So exocytosis is a piece leaving the cell, large piece. It does require energy to do that, so some ATP would be used. So there's a couple of different types of endocytosis. This is bringing it in. You can see again these projections coming out. This would be food coming in. It could be other nutrients coming into the cell, molecules coming into the cell. Do you need to know all these different types? I would say no. But just know that this is bulk transport through endocytosis. I would be aware of that term, endocytosis. And then here we have receptor mediated. So this just means you have to have the receptors met, and then that vacuole would be, or that vesicle would be created from there. So that leads us to this. Now, normally I would do this. Right now I can't really give you candy, but if you look at the joke here, what you got there, Jim, transport proteins. So he eats transport proteins, and now he can leave the cell. He was in a prison cell. He's leaving the cell, transporting out of the cell. Ha, 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 ha. I know you're laughing. And that is it for Chapter 7. Transport